Well, it's time to start our lecture on Chapter 14. The focus is on erythropoiesis, which is the synthesis of red blood cells. We will also focus on red blood cell removal, and I will also talk about the various types of anemias. Regarding blood typing, which is also covered in this chapter, that should have been covered in physiology. I will upload the lecture that I give on blood typing, and I will also give you a link for a podcast so you can listen to it if you want to review it. It also, in that lecture, it covers hemolytic disease of the newborn, or HDN. I highly recommend that if you've never heard of HDN, then you should listen to my lecture on blood typing. There is a lot to cover in this chapter. It's a small chapter, a short chapter, but nonetheless it is very dense with a lot of good information. I'm going to review properties of whole blood and the components in blood, and then I'm going to move into red blood cell indices. We've already covered these, of course, like what's MCH, what's MCHC, etc., etc. And then I'm going into how blood is made, how we actually assemble the hemoglobin, I should say. And moving on, I'll discuss some red blood cell pathologies like polycythemia, and in the last few segments I'll cover the anemias. You probably know this from physiology or even from your anatomy class. Blood is going to transport gases like oxygen, carbon dioxide. It will also transport nutrients, hormones, waste, anything that can be dissolved in the plasma. Blood is important for maintaining blood volume. It is also important for maintaining blood pH, and it's very important for maintaining the body temperature. Remember, most of the body's core temperature is shunted to the most internal regions of the body. Blood is also important with maintaining electrolyte levels, and we also recognize from our last chapter that blood is very important for maintaining hemostasis. That means the cessation of bleeding, when appropriate, of course. And we also know there are white blood cells in blood that are going to help protect us from pathogens. There are more liters of blood in the adult male than the adult female. On average, there is going to be about 4 to 6 liters of whole blood in a given individual. Again, males will have more than females. The average temperature of blood is about 100 degrees Fahrenheit. And it is true when we say that blood is thicker than water. Blood is about 5 times more viscous than water. And can you tell me what makes up the viscosity of blood? Well, it is the most abundant formed element that makes up the viscosity or contributes to the viscosity of blood. That would be the red blood cells. If a patient makes more red blood cells than normal, a condition known as Polycythemia Vera or secondary polycythemia, their blood viscosity is going to increase, and this can increase the workload of the heart. A patient with anemia who has reduced red blood cells, well, that can decrease the blood viscosity. Blood pH ranges from 7.35 to 7.45, with the average being 7.40. On either extreme, we can have changes in the pH causing denaturation of plasma proteins. Blood is going to change its color when it's oxygenated, or 100% saturated, as we say. It's very bright red, as you can see on my little color swatch here. When it's oxygen poor, it looks more of a darker color. It's not blue, like a lot of students think. The cyanosis that you see associated with poor oxygen content in a patient is really from the pigments in the skin. Blood without a robust amount of oxygen is going to be more of a burgundy color. We've seen this slide before. Whole blood is a mixture of plasma and formed elements. The formed elements, most of them, are red blood cells, of course. Platelets and white blood cells are going to be less than 0.1%. In the plasma, we see it's mostly water. About 90% of it is water. Plasma proteins, the most abundant of which is albumin, is found in plasma, and other solutes can be dissolved in plasma. In this picture shown here, I'm showing you a healthy picture of red blood cells and various types of white blood cells. You can see an eosinophil, and and you can see a lymphocyte. I see a monocyte, a neutrophil, and a basophil. All of them are there. Plasma is mostly water, as I just said, which is needed to transport all of the solutes that are dissolved in the plasma. There are organic and inorganic materials. The plasma helps retain the heat of the body. Water has a high specific heat, so it can absorb a lot of heat before it rises in temperature by 1 degree, which is the reason why our blood is important in regulating body temperature. What is dissolved or even suspended in plasma? There are proteins, about 7 grams percent plasma protein content. That means 7 grams of proteins within 100 milliliters of whole blood. We see most of this comes in the form of albumin. Albumin is very important for regulating osmotic pressure. We saw this with the example of kwashiorkor with a young child who was on a low protein diet or protein deficient diet. That child was not making sufficient amount of albumin, and therefore the osmotic pressure in her blood compartment was reduced compared to her transcellular compartment. And that's the reason why we saw the ascites in the peritoneal cavity. The albumins can be non-specific transport proteins. They can bind to hormones and other lipids to help transport them throughout the body. The other types of plasma proteins are globulins and fibrinogen, which of course we just learned in our hemostasis lecture about fibrinogen, of course. There are other regulatory proteins like hormones that can be dissolved in the plasma. Inorganic solutes will include electrolytes like calcium, magnesium, sodium, which is the most abundant cation and is the most involved in maintaining osmotic pressure in addition to albumin. There are organic nutrients like carbohydrates and lipids, and some organic wastes are also found in plasma. Organic wastes include urea, which is a product of protein metabolism, uric acid, which is a product of nucleotide metabolism, creatinine, which is a result of protein metabolism. Bilirubin, you will soon see, is a result of red blood cell destruction, and ammonium ions, which are a result of protein metabolism. Maybe this is new for you, or maybe this is review, but where we have hematopoiesis or synthesis of red blood cells, specifically the erythropoiesis, I should say, the locale shifts throughout our development. In the early months, the embryonic months, hematopoiesis is occurring in the yolk sac, as you can see here. And then as the fetal period begins, we see a transition from the yolk sac to the liver and spleen, primarily the liver. And then before birth, around there, we are going to again see a shift in locale, and this time it's to the bone marrow, the medullary cavities of the long bones specifically. But as you reach, well, a little bit prior to age 20, we're going to again see a shift in the locale of hematopoiesis. We're going to lose the red marrow in our long bones. The medullary cavities of the long bones will be filled with yellow marrow, which is fat. And you see there's a shift from the appendicular skeleton or long bones to the axial skeleton. In other words, hematopoiesis is going to occur in the vertebrae, in the sternum, in the ribs, and even a little bit in the iliac crest. So it's all downhill after age 20, folks. When we speak of erythropoiesis, we are starting with the hemocytoblast, which is a multipotent stem cell. Multipotent means that it can replicate and divide and give birth to many different types of daughter cells. The first branching from this hemocytoblast cell will either be a myeloid stem cell or a lymphoid stem cell. The myeloid stem cell can give birth to daughter cells that will ultimately differentiate into erythrocytes, or granulocytes like eosinophils, neutrophils, and basophils, or monocytes that will differentiate into macrophages, or the megakaryocyte, which of course we already learned, which generates platelets. The other type of stem cell line is the lymphoid stem cell line, which will propagate T and B cells. So we say multipotent again to refer that the hemocytoblast is determined, meaning it already has selectively turned on genes and turned off other genes, and can give birth to many different types of daughter cells. But as we go from left to right in this picture that we see on the screen, there is going to be progressive differentiation. What exactly is the recipe for erythropoiesis? Well, we need, of course, to start with a healthy stem cell. We need healthy hemocytoblasts. There needs to be chemical signals that promote the daughter cells to progress from the hemocytoblast to the myeloid stem cell lineage, and to actually go through the phases of erythropoiesis, to go to a proerythroblast, to a basophil erythroblast, etc., etc., to a reticulocyte, to an erythrocyte. We need good growth inducers like interleukins, which are cytokines. We need differentiation inducers, again, various types of cytokines. They are all chemical signals released by adjacent cells to help direct the hemocytoblast to generate daughter cells, etc. We also need good, healthy cell division. The hemocytoblast needs to replicate its DNA and then divide. There needs to be healthy amounts of nucleotides to promote DNA synthesis, because the S phase in mitosis, or the mitotic process, precedes the actual mitotic phase, or M phase. That means we need good DNA replication, and in order to do that, the enzymes of DNA replication require vitamin B12 and folic acid as coenzymes. And I will review what a coenzyme is in a future segment when I start talking about anemias. Obviously, if erythropoiesis is to occur properly, there needs to be good hemoglobin synthesis. I'm going to talk about assembling the components for hemoglobin. I'll talk about the synthesis of heme, and I'll talk about iron again. I'll also talk about the globin chains and their gene expression, and how we have different types of globin chains throughout our development.